It's an absolute pleasure to welcome Greg Forrest from the Georgia Institute of Technology. I keep mixing that up. Greg studied mechanical engineering at the Massachusetts Institute of Technology in Boston. He got his PhD there in 2007. He then moved to Georgia Tech in 2008, starting there as an assistant professor at the Woodruff School of Mechanical Engineering, and was made a full professor in 2019. Based there, he's been focusing on the creation and application of miniaturized, high-throughput robotic instrumentation with the aim of advancing biomolecular science. And part of that, of course, is developing the fundamental engineering that makes such instrumentation possible. He and his lab have been focusing on automating patch clamp recordings, among many other things, and for quite a while now. And probably the biggest step towards making that possible and really pushing this is the development of pipette cleaning. And that's what he's going to be talking about for the next 30 or so minutes. And then, as I said before, if you have any questions, drop a message, raise your hand, just let us know, and we can do a question and answer afterwards as well. Thanks, Greg. I'll hand over to you now.
Well, thanks for the nice introduction, Christian. Thanks for the opportunity to be here and share with you. And I'm happy to take questions along the way in the chat and share with you some of the discoveries we've made over the past few years on patch clamping. So let me share my screen here. There we go. Okay, I'm going to make this full screen. We all set there, Christian?
Yep, coming through fine.
Okay, great. So the title of the talk here is Cleaning Patch Clamp Pipettes for Immediate Reuse. And so I'm talking about these kind of glass micro pipettes that we all know and love. Can you guys see these? Can you see me still?
Yeah, small, but yes.
Yeah.
Yes.
So, you know, patch clamping is a Nobel Prize winning technique. It's been around for decades. And in the Nobel Prize winning speech, Hamill remarked that each pipette should be used only once. You know, when you patch clamp onto a cell, you take a fresh pipette, as I'm sure you know, you touch it delicately to a cell's membrane and apply a bit of suction to form a seal there, typically over 1 gigaohm in resistance. And then you apply a bit more of suction to cause a small hole in that membrane. And in doing that, you can imagine there's debris, you know, from the bilipid layer of the membrane, which can stick onto the pipette, either on the outside or on the inside. And here's a picture of what it actually looks like. Here's a brand new pipette, freshly pulled. This is imaged, you know, down the barrel, looking at the very end on version of the pipette. This, by the way, this little bump here is not some dirt. That's actually the filament inside the pipette, which a lot of people use filamented pipettes. And after patching, oh my gosh, look what happens to this thing. It's got debris all around the glass face as well as on the inside. And as you can imagine, that would make it very difficult to seal against a second cell. So that's why Hamill says, just make sure you use them only once, make sure they're freshly pulled. And so this was the dogma for about 30 years. And, you know, at places like the Allen Institute, where they published a paper recently where they patched about 22,000 cells. And I was there for some of that work in person. I worked there for about 8 months, and I can tell you with certainty they used a fresh pipette for every single one of those recordings. So we were so surprised to discover back in 2016 that if you dunk the tip of the pipette in Alkanox, which is like a detergent and surfactant that we all have in our labs already, it comes out just like new. Look at that beautiful tip of that pipette. So that's what I'm going to share with you some of the details of over the next 25 minutes or so. So just to be clear here, manual patch clamping, as it's done by about 5,000 people around the world today, you install a pipette, approach the target cell, gigaseal against it, just like I've shown you in the cartoon on the left here. Then you do a whole cell recording, and then you replace the pipette. And if you're really good, if you've done this many, many times, you can replace that pipette in 1 or 2 minutes. And so someone like this guy, Bo Young here, this is Bo Young, who's personally patched about 5,000 cells in his lifetime. I think he might be one of the world record holders. He can do about 8 to 10 cells a day. He's really good. Now, there's 2 variations that become possible when you've got a ability to clean and reuse those pipettes. One is, after you do your whole cell recording, you push a button, and the pipette comes back cleaned in 30 seconds. That's one option. That would be useful if, for example, if you're doing manual patch clamp recording. So you still want to drive the pipette to the cell. You still want to do the giga sealing the way you want to do it. You still want to do the recording the way you want to do it. It's only after the recording that you engage some kind of automated system to clean and reuse that pipette. And then there's a sort of fully automated system. We've been calling it the patcher bot, like robot, right? Patcher bot. And here you approach the cell, giga seal and whole cell recording, and then it cleans and returns. And all these blue squares are automatic. So it just goes around and around and around and around. And that way you can do about 10 cells per hour instead of 10 cells per day. Okay, so this is what we have working in my lab on multiple rigs. And this has now been shared with many collaborators around the world and many customers around the world who are down doing this. So if you've got a scientific goal that requires hundreds or thousands of recordings, I strongly encourage you to clean and reuse pipettes to try to make it faster. 10x increase really makes a difference in the kind of scientific questions you can ask. The bottom line here is pipette cleaning enables serial automated patch clamp recordings. Now, in case you don't believe me, I got a cool video. Let's play the video here of what this thing looks like in action. Now, this is on a Scientifica rig. We've now done it with Sensipex. We've done it with Luigs and Neumann. We've done it with, everybody's Sutter. It's not about the actuators. It's about patch clamping as normal and then reusing the pipette. So on the left, you see the rig. It's been programmed so that this green light comes on when it obtains a whole cell. You see the green light flashing in the lower left corner? It does that when it's got a whole cell. On the upper right, you can see a screenshot of what the camera sees from above. So this is patching in hex cells here. Neurons in culture, I'm sorry. Well, it says neurons, but I think those are hex cells actually. And then we've pre-marked the locations. Those red circles are the targets. And then it goes and gets them one by one. And there's nobody touching the rig at this point. So I want to take you just down a little memory lane here of how we actually did this and what we saw, like the first time we ever saw it. And you can ask your questions and be surprised along with us. What happens on the end of that pipette is you get this kind of junk. This is what a cell membrane looks like. It's got fats. It's got sugar molecules. It's got proteins. It's got all kinds of stuff. And what we found is that initially Alkanox is what we were using. And I'll tell you a little bit later how we even further improve that. This is a surfactant and a detergent, and it basically cuts and rinses away those molecules. Here's the procedure that we figured out the very first year. We move to the cleaning basin. That takes about 10 seconds. We suck up a little bit of cleaning solution at negative pressure, negative 345 millibar. So this has to be digital pressure regulators on here. You don't want to do this with your mouth. That would just be, you'd have to be very patient. But you basically go in, out here 5 times. And then at the very end, you flush it all out. And this flushing for 5 seconds, we calculated that flushes out like 100 times more recording solution than you ever sucked up in cleaning solution. So there's no cleaning solution left in there. Then you move to a rinse basin, which we see here, rinse basin, and you again suck up and squirt out once, and then you're back to your preparation. So that took us 60 seconds. Now, let me take a step back here. So let's look carefully at what happens when you're patch clamping. Here's what it looks like in human embryonic kidney cells or HEX cells, and here what it looks like in a mouse neuron embedded in a slice of brain tissue. Either way, you're going to have this characteristic increase in resistance as the pipette's face seals or tip seals against the cell membrane. So it increases gradually over the course of maybe a minute or two at most to get up way over 1 gigaohm. And anything over 1 gigaohm is considered a giga seal, and you're good to go for your next step, which is the break-in process. So we measured the time to giga seal, and we measured the resistance at the maximum giga seal resistance to see if this cleaning was really working. So you start with a fresh pipette. Of course, you see this characteristic increase. Now, if you wash the pipette in ACSF, just artificial cerebral spinal fluid, if you do the same exact cleaning procedure I just showed you, okay, let me just jump back here. You do the same exact procedure on the right here, okay? But the pipette is in ACSF instead of Alkanox. It's not reusable at all, okay? You cannot obtain a gigaseal again. And that's already been well known. I mean, that's why Hamel mentioned it in his Nobel Prize winning speech. Now, some people thought bleach works. There was a suggestion back maybe 5 years before us that bleach can clean patch clamp pipettes or patch chips. We did not find that to be the case. We were very disappointed. We thought bleach was a great idea. We tried a whole bunch of other things like organic solvents, all kinds of things. It wasn't until we found Alkanox that we got here another gigaseal in about the same amount of time. Now, this is just one data point, so let me show you a lot more. Again, Alkanox, very easy to buy. So here's fresh pipette gigaseal gigaseal resistance versus ACSF versus bleach, Triton X, acetone, PLA, Alkanox, and as you can see, only Alkanox had the difference that was not statistically significant. Now this is with large N here, okay? So this is a distribution, and it's no difference. This is using a one-way ANOVA. And so, again, looking at it with the scanning electron microscope, the fresh pipette tip versus the one that's been used and cleaned with ACSF. This has been cleaned with ACSF, doesn't make it clean at all, and then with cleaning with Alkanox after. So we were wondering then, of course, how many times can you do this? So here we've got the fresh pipette is the very first one, and then the first cleaning would be the second recording, right? And then the second cleaning would be the third recording, and so on like that, all the way up to the 10th cleaning. And what we see here is both in the gigaseal resistance as well as the time to gigaseal, there is no effect of the number of cleans. You cannot see any statistically significant change in the gigaseal resistance or the gigasealing time. So that was pretty exciting. We were pretty excited about this. Now, we thought, okay, now I don't know if I trust this. Maybe there's some residue in there somewhere, right? So we, here at Georgia Tech, we're fortunate to have a lot of instrumentation. So we happen to have a $1 million electro spray ionization mass spectrometer in the basement of this building I'm in right now. So we take the pipettes down there. And this thing is capable of seeing almost single cell molecules, okay? And so we have the pipette after the first reuse and the 10th reuse, and we don't see any signs of the characteristic, this molecule here, sodium alkyl benzene sulfonate, LAS, that is one of the ingredients in Alkanox. Now, it's a proprietary formulation, but that's one of the main ingredients. And we see, look at that, on the mass spec, some of the fragments of that molecule clearly standing up here, and we did not see any of those on the pipettes that had been used for cleaning. That showed us that the rinsing procedure and the process of expelling the cleaning solution from the pipette was working, at least as far as any mass spec could see, which is the most sensitive way of detecting single molecules. But what about an even more sensitive test? How about single ion channel measurements? These are notoriously difficult to do with patch clamping. The currents are in the single nanoampere range, okay? Is it possible that cleaning would change the way that individual ion channel currents can be measured? We perform these measurements as rigorously as possible with our collaborators on hex cells that were transfected with certain ion channels. And we deliver a drug, GABA, and see the current through the ion channel. This is a single ion channel recording. And again, we see no difference in the current between a fresh pipette and a pipette on the fourth cleaning, okay? And here's all the data here in terms of the characteristics, the max current, and so forth. These are some of the metrics that people care about when they're doing single ion channel recordings. So this is the most sensitive type of patch clamp recording that can be done. And we see no difference in the biological measurement. Another way we check to see if this is working is we did patch clamping in brain slices, and we put a dye inside of the pipette so we could later reconstruct the morphology of the cells. So this is a technique where patch clamping can reveal both the electrophysiology and the morphology for structure function studies. A lot of people are interested in this kind of thing. So here's a slice of brain tissue, neuron 1, 2, 3, 4, 5, 6, 7, 8, all patched with the same pipette. One pipette patched all of these cells. And we see that there was no change in the morphology from what people have come to know from these types of cells in these brain regions. So let's talk more about these slice recordings, because I showed you hex cells, and that's cool. But what about brain slices? Brain slices are a lot more difficult to patch in, as you may know. You know, in hex cells, you can get 70, 80, 90% yield. In brain slices, you get maybe 50, 60% yield. So what I'm showing you now is in 16, 17, 18 minutes on this recording, this robot has obtained 5 whole cells out of 5 attempts. And what you're seeing here is the fastest throughput of whole cell patch clamping and brain slices ever performed. Remember, I told you 8 to 10 cells a day for a good patch clamper. Now this is 30 minutes. And this is in brain slices. This led to the funding of a $3.8 million R01 grant with Matt Rowan at Emory to do this in the brains of mice with Alzheimer's. But just to show you the power here, I just showed you a video of automatically performing 8 whole cell recordings out of 10 attempts in 41 minutes in brain slices. Here they are. Here they are, how they appear in space in the brain slice. The 8 green ones. Of course, it's not perfect. There were 2 failed attempts. But we did a series of current injections, all automated. It's all automated, 100% automated. We've done it in the hippocampus. We've done it in the thalamus. In case you had some doubt that maybe this technique doesn't work in different brain regions, we do not find anything different about the statistics, the gigacil resistance, the series resistance, the baseline membrane potential. None of the quality metrics are affected by cleaning. We've checked all those. So this rig here, just so in case, just get you make sure fully oriented, we've got the indicator light. We've got 2 manipulators now on this one, 1 and 2. We've got a microscope above. We've got this timer, the whole cell counter. And let's watch this thing in action. Okay, this is Corey Landry setting this rig up. We've got the clock here, so you can see that it's real time, what's going on. Of course, we sped this up, but you can see the clock, so you know we're not cheating. What he does is he's selecting about 10 cells, and then he goes and gets a coffee. And so this rig just runs all day with 1 pipette. Isn't that amazing? What Corey achieved here is 10 cells per hour, eliminating bias in his experiments. Of course, every cell has the exact same approach strategy. It has the exact same pressures applied to it. It has, the exact same criteria for break-in. And so we did some blind experiments with Corey where he didn't know what was in the bath. We did some with saline, we did some with Alkanox, and we started experimenting with this other chemical called tergazyme. Now, tergazyme is an improvement upon Alkanox. It has an enzymatic agent in it, so it cleaves proteins as well as a detergent and surfactant. And lo and behold, we found unbelievable throughput. Let me jump ahead, I'll come back. But I want to show you some of Corey's data. I'll jump back in a minute. Just bear with me. Look at this. One pipette, 102 whole cells in 123 patch attempts with an 83% yield on 6 HEC 293 cover slips in 12.5 hours. As far as I know, this is the most number of whole cells ever recorded in one day by one person, with a single pipette. There's every single recording right there, in case you had any doubt. Pretty cool, huh?
Mike has a question. Sorry, I noticed this for a bit, but I was waiting for a break.
Yeah, Mike, what's up?
So thank you very much. Can you hear me?
Yep.
Mm-hmm.
Okay, great. Thanks so much for making this presentation. It's fascinating, actually, and this latest data is quite impressive. I mean, basically it's a game changer if you really think about it. But the question that I have goes back to the cleanliness of the pipette after the cleaning, and whether there's any residual on it. And I'm wondering if that's a bit of a red herring, because when you put a fresh pipette in anyway, I mean, I've seen some of the labs that these rigs reside in, and it's not exactly the most cleanly environment. Isn't there already stuff, grunge on the pipe, a lot of the pipettes anyway? Am I making myself clear?
Right. Well, in a normal laboratory, you could think of it as a class 100,000 clean room. What that means, of course, it's not clean at all. What that means in a class 100,000 clean room is in a cubic foot of air, there's 100,000 particles. And some of these are charged, right? And so it's well known that, from the images from Sutter, the Sutter pipette handbook, that things will stick to the pipette over time. That's why they say the pipettes have to be fresh. You can't just pull 100 pipettes and then use them all throughout the week. You have to pull them each morning that you're going to be patch clamping. And so, yeah, people do patch with pipettes with debris on them. All I can say is the proof is in the pudding. We are not seeing any difference in the amount of time we can hold the cell, in the currents from the cell, down to the single ion channel level, and even over 100 cells. You would think if there was some contamination here, it would start to eventually give out.
Well, that's what I'm saying. I
right.
mean, that's the point I'm making, is that it seems to me that people worrying about contamination from the fluid it's a bit overblown, don't you think?
Right. And then to put a finer point on it, we were stunned when our colleague Geiger, I'm going to jump ahead here to make this point a little bit more clear. There's a colleague in Germany, Jörg Geiger, and this is some work out of his lab. This is not our work, okay? So what he did was he built a rig with 1, 2, 3, 4, 5, 6, 7, 8 manipulators. And he was hoping to get a connectomics study where, in connectomics, you want as many simultaneous patches as possible. So if you go in with 8, and the Allen Institute did a similar study, if you go in with 8, you may only get 4 or 5, or if you're really lucky, 6 whole cells out of 8. And that significantly limits the number of connections that you can probe, because the number of connections goes with the square of the number of manipulators. So if you only get 6 instead of 8, you only have like 36, actually not 36 minus 6, 30 possible connections. Whereas if you get all 8, you got now all of a sudden 64 minus 8, 56 possible connections. A significant improvement. 56 is much higher than 30. So you can see why Jorg jumped at this and said, if I can clean and bring those 2 failed pipettes back into the picture and try to get those on cells too, the matrix, the square matrix, gets much bigger. So this is what he did. And he showed, for the first time, probing up to 600 synaptic connections in human brain slices in one day. Okay? By cleaning and re-attempting to patch with the pipettes that did not patch the first time or the second time. So in this way, he was able to get a full matrix, 8 by 8, every time. Brilliant. And here's the coolest thing about it. Are you ready? You want to talk about...
You're not showing the script. Sorry, you dropped out of the presentation.
Oh gosh.
No worries.
I'm so sorry. Let me fix that for you. Thank you for interrupting me. Okay, so here's the slide.
Get back.
Here's the slide. You see Jorg's rig here, the schematic and the picture of it. You can see that now, Christian?
Yep.
All right. So we were just talking about contamination, and the point I wanted to make here is what Jorg discovered, going beyond what we had discovered. He had this sentence in his paper: the final expulsion sequence does not necessarily require additional wells containing ACSF. In other words, he's not rinsing his pipettes. And the way he explained it in his paper, again, this is not our work. I want to be respectful of his work. But when the pipette goes back into the to patch another cell, of course, it must first pass through a fluid layer where the media is flowing over the top of the slice. And he said that rate of fluid flow is so great that it rinsed the pipette as they descended towards the slice. Before they even touched the slice, the pipette was completely rinsed. So we actually started doing this. I don't know if you know that, Christian. In my lab, we don't even rinse anymore.
Yeah, you mentioned it. It's very cool.
So this was an awesome discovery. And so again, he showed in this paper thousands of connections with no deleterious effects from cleaning. So, I mean, what more can people say? That it works in different cell types, it works in different brain regions, it works at the single ion channel level, it works at the whole cell level, it works for connectomics, it works for pharmacology, it works for disease studies. If contamination is real, I would think we would have seen it by now. So here's our PatcherBot performance. This first column is hex cells, and then the second column is neurons and brain slices. And in both cases, we can walk away now for about 45 minutes on average. And this is over many hundreds of recordings. Average time per attempt is about 4.5 minutes with hex cells, 3.9 minutes with brain slices. Number of cells we pick is typically around 10 now, or in brain slices 7, because the brain slice is a little squishy, and so cells tend to move around a little bit on you. So we normally keep it to about 7. And then this is the worst success rate. In our lab, we try to be open and honest about our success rates. We don't report experiments that weren't successful. We report all experiments. We don't not report. I said we don't not report experiments that weren't successful. We report all experiments. So our worst success rate was 6 out of 10 in this data, and 3 out of 7 on the cortex, and 1 out of 5 on subcortical. And our best experiments in both cases, 10 out of 10 in the slices. I'm sorry, in the hex cells, and 6 and 7. 6 out of 7 and 5 out of 7 on the neurons. So pretty good. So here's the dual patcher bot. Of course, once you've got one automated, now you can add a second one. So this is that picture I showed you earlier, but now it's a video. And the timer is minutes, so it's 6 minutes, 7 minutes, 8 minutes. And these pipettes are taking turns now, taking turns. So one is cleaning while the other one is patching. They're taking turns. And there's nobody in the chair. So we can now do, you know, a whole PhD's worth of work in about a month with this. So I have the paper right here in front of me.
Okay.
This is the paper right here.
Yeah, exactly.
And it just came out this year. So I just read the paper for the first time. And listen, I think, you know, the more we can automate patch clamping, the better. But that being said, I see there's a section in here where they tried to do in vivo recordings and they required Turgozyme. Ultrasonic cleaning in Turgozyme allowed the reuse of the pipette. This is a line from the paper. Ultrasonic cleaning in Turgozyme allowed the reuse of the pipette. So I don't know what the contamination concerns are. Like, I'm reading a line from the paper right now that says, even if not detected, there might be some trace left of these chemicals inside the patch clamp pipette that can be harmful to the cells. I don't see any evidence of that. I don't know what to make of that sentence. I haven't seen any data to that to suggest that. What I know about the enzymatic cleaning is it works for more than 100 uses. I know it works in these different types of cells. I know it works in vivo. So this is, let me show you this video. See if this plays. Is this playing for you guys? Can you see this?
Yes.
Okay.
Yep.
This is in vivo patch clamping now. So do you see in the lower right corner is the mouse's head with a craniotomy? Can you see that?
Yes.
Okay, so here we are showing the pipette is being reused for in vivo patch clamp recording. And let me reread this sentence out of this paper from Luigs and Neumann. Ultrasonic cleaning in Turgozyme allowed the reuse of pipettes. This video is showing this video is showing reuse of pipettes without ultrasonics in Turgozyme. So it's identical, except there's no ultrasonic.
Okay.
Okay. So at the very least, the data I'm seeing so far is not for 100 reuses. It's not in vivo. It appears that there's some limitation. I don't understand it completely. The data is not complete right here, what I'm looking at. Now, maybe in a few years it will be better. You know, that would be great. Like, you know, the more the merrier. Let me show you one last cool slide here to kind of wrap up the data part of the presentation. This is an experiment where we did all day patching with a single pipette on the left. And the yield was tremendous. You know, this dotted dash line represents a perfect yield of 100%. So if you had 100 attempts and you got 100 cells, whole cells, that would be this dash line. And here it's just slightly below, about 80, I believe 80, 85%. And here it is with, this is doing an optogenetic studies. So these hex cells are transfected with channel rhodopsin 2. And when the light comes on, it induces a photocurrent here. And here you can see the number of cleans, 8, 22, 27, 34, 44. Look at that, we're getting the exact same photocurrent after 44 cleans. And this is now up to 90-something% yield, right? So this is about a 4-hour experiment, patching 50 cells in 4 hours. So you know, that's what I'm excited to share with you today, about the cleaning. It works. It definitely works, and it definitely enables automation.
Okay.
Thanks, everyone.
So thanks for your time. Thanks, Greg. That was a great presentation and lovely discussion afterwards.